So welcome to the last lesson in the biochemistry special topic, stereochemistry in biomolecules. So by the end of the lesson, you should know that most amino acids are chiral. Naturally occurring unsaturated fats is mostly in the cis form. DNL stereoisomers in sugars and what they actually refer to. Ring forms of sugars have isomers, and vision chemistry involves isomerism in some way as well. Going to have to be able to describe the hydrogenation of unsaturated fats, explain and the structure and properties of cellulose, and discuss the importance of cellulose as well. And then finally outline the role of vitamin A in vision. Let's get to it. So isomers, this is pretty much a review of organic chemistry. So most biochemical processes are stereospecific. They involve only molecules with certain three-dimensional configurations, and molecules that have the same sequence of atoms and chemical bonds but different arrangement in atoms are known as stereoisomers. Stereoisomers that cannot be transformed into one another without breaking a chemical bond are known as configurational isomers and include the two classes of cis and trans and optical isomers. And both of these stereoisomers play an important part in biochemical reactions. So first, let's look at the amino acids. So proteinogenic two amino acids, okay, those that form proteins, are relatively simple biomolecules with the general formula previously outlined. With a single exception, all two amino acids have four different constituents: the amino group, the carboxyl group, an R side chain, and a proton, attached to the same carbon atom, C2. Okay? These molecules are therefore chiral. They can exist as two enantiomers, which are the mirror images of themselves. In, for example, glycine, R equals H, so the C2 atom has two hydrogens and is thus not chiral. All other amino acids are exclusively, proteinogenic amino acids, those that form proteins, are exclusively L isomers, though several D amino acids occur in bacterial cell walls and antibiotics. Now you'll notice, of course, that the three-dimensional structures of chiral molecules can be represented on paper using the wedge and dash notation, or as Fischer projections. Now, in the wedge and dash notation, as we see here on the slide, any two of the four chemical bonds formed by a tetrahedral carbon are positioned parallel to the plane of the paper and drawn as plain lines. The third bond coming out of the page towards you is represented by a wedge, as the fourth one coming away from you is represented as a dashed wedge. Okay, this is where I use the Korn rule as well. You place the hydrogen facing away from you, okay, so it's up. If the C double bond, or the carboxyl group, the R group, and the NH2 groups are spelled clockwise in a D enantiomer, and anticlockwise. And if anticlockwise, it is an L enantiomer, the C, O, R, and N. Yeah. Okay. The other way you can represent amino acids is through Fischer projections. Okay, so they're known as Fischer projections. Okay, and are designed to represent the stereochemical formulae with plain lines, which is particularly useful for complex molecules with many stereochemical centers. In Fischer projections, the carbon chain of the molecule is drawn vertically. Okay, so we'll just use a simple one, say like alanine. Okay, so there we go, got this carbon chain there. With the senior substituent, so it would be the carboxyl group as the senior substituent in amino acids, drawn at the top, okay, and the chiral carbon is shown as a cross, okay, so the center of the cross will be your chiral carbon, and the horizontal lines represent the chemical bonds coming out of the page, and the vertical lines representing those pointing away from the page. Okay, so if we had NH, my writing is terrible with this pen, we have NH2 here and an H here, and the only thing left is methyl for alanine. This would be the example of L alanine. Okay, and therefore for D-alanine you just swap these here. Okay, so that would become NH2, and that would become hydrogen, and that would become D-alanine. In Fischer projections, the L enantiomers of two amino acids have an amino group on the left of the chiral carbon, while D enantiomers have it in the opposite orientation. In order to identify a particular enantiomer, a wedge-dash formula or a three-dimensional model of a chiral compound, you can use it using the Korn rule, which I've already discussed in the slide. Okay, so in terms of fatty acids and triglycerides, naturally occurring unsaturated fatty acids have a cis configuration of a double bond, carbon-double bonds. The hydrocarbon chains in such molecules cannot adopt linear conformations, which prevents them from coming close to one another and reduces the intermolecular forces. As a result, cis unsaturated fatty acids and their triglycerides are often referred to as cis fats and are usually liquid at room temperature. So hydrogenation of vegetable oils is commonly used in the food industry to produce saturated fats with high melting points, margarines. The reaction of unsaturated triglycerides with hydrogen is similar to the hydrogenation of alkenes and takes place at high temperatures in the presence of a nickel or platinum catalyst. Hydrogenated vegetable oils are cheap and offer many benefits, including the absence of cholesterol, has a controlled texture on the mouth, spreadability on your bread, increased resistance to heat, and extended shelf life. Now, of course, with everything, there's always bad parts. The high temperature of hydrogenation also partially converts the cis isomers to their trans isomers, which the body doesn't particularly like. Okay, and if the hydrogenation is incomplete, the final product will contain trans unsaturated triglycerides, known as trans fats. In contrast to their cis isomers, the hydrocarbon chains of trans fatty acids and their triglycerides are less distorted and can adopt a more rod-like conformation. Therefore, they tend to have higher melting points than their cis fat analogues. Now, although trace amounts of trans unsaturated fats are present in all natural products, the higher levels of trans fats associated with partially hydrogenated oils are quite high. For example, 15% in margarine. Due to their increased heat resistance, partially hydrogenated oils are widely used in the food industry. However, excessive consumption of trans fats is linked to high LDL cholesterol levels, which increases the risk of coronary heart disease. And also, these fatty acids take longer to metabolize than their cis counterparts, so they accumulate in fatty tissues and increase the risk of obesity, which nobody wants. Yeah. That's kind of what I've just already said in the terms of their advantages, in terms of shelf life and spreadability. Okay, so in terms of the carbohydrates, only D forms are actually found in nature. In the solid state and in aqueous solutions, monosaccharides exist predominantly in cyclic forms, which are commonly represented by the Hallworth projections, which we've previously touched on. Each monosaccharide can produce two cyclic forms that differ in orientation at the OH group of the C1 atom of aldoses and the C2 of ketoses. In alpha glucose, the OH group of C1 lies below the plane of the ring, while in beta glucose, the group lies above the ring. Similarly, alpha and beta isomers of fructose differ in the position of the OH group directly attached to the C2 atom. Here we go, C2, see? Beta above, alpha below. In aqueous solutions, alpha and beta isomers of monosaccharides can transform or interconvert between each other via the open chain form, which exists in equilibrium with both cyclic forms. For most monosaccharides, beta isomers are slightly more stable than alpha forms. So, for example, in glucose, it would basically be 64% alpha and 36% beta, and about 0.3% in the open form. So, in terms of retinol and vision chemistry, the collective name vitamin A, which we looked at previously, refers to a group of polyunsaturated compounds. One of these compounds, retinol, which we've also looked at, is a long-chain aldehyde involved in vision chemistry. In the photoreceptor cells, retinol exists in two stereoisomers, the cis and trans, which can be interconverted into either by the action of visible light, which we see in the diagram. The aldehyde group of the cis retinol can reversibly bind to the lysine residue of the protein opsin, producing a light-sensitive pigment called rhodopsin. The C double bond N bond of rhodopsin extends the system of electron conjugation in cis retinol and allows it to absorb visible light in the blue and green regions of the spectrum. As a result, pure rhodopsin has a reddish purple color and is often called visual purple. Other proteins of opsin family produce cis-retinol complexes with different absorption spectra, which is responsible for the color vision associated with us humans and other animals. Okay, and that's how the conversion occurs. Okay. When rhodopsin absorbs a photon of visible light, the residue of cis-retinol isomerizes to the trans. Okay, you see in the red there. And a conformation changes, the shape changes of the protein, triggering a cell response that eventually sends an electrical signal to the nervous system. At the same time, transretinol detaches from opsin and undergoes a series of transformations known as the visual cycle. At the end of the visual cycle, transretinol is converted back into cis-retinol, which reattaches to opsin and produces a new functional rhodopsin complex.